Hello everyone, my name is Rahul Gupta. I am B.Tech's 4th year student. My roll number is 56 and my group is 72. And my topic is amino acids and proteins. So first introduction. The 20% of human body is made up of proteins. Proteins are large complex molecules that are critical for normal functioning of cells. They are essential for the structure, function, and regulation of the body's tissue and organs. Proteins are made up of smaller units called amino acids, which are building blocks of proteins. They are attached to one another by peptide bonds, forming a long chain of proteins, like the like in this example. So, amino acid structure and its classification. An amino acids contains both a carboxyl group and amino acid group. Amino acid that have amino group bonded directly to the alpha carbon and referred to as alpha amino acid. Every alpha amino acid has a carbon atom called alpha carbon. So these are C alpha, so bonded to a carboxylic acid. COOH group, yani oxalic group, and amino group. NH2 is a hydrogen atom, and R group that are unique for every amino acid. Like these are there are four different points uh, attached to the carbon, like hydrogen, NH2, R, and HOOC. Like H is alpha carbon, C alpha amino group, NH2, R group various of the every amino acid is called R, and COOH carboxylic acid group. Classification of amino acids. There are 20 amino acids based on the natural of their R group. They are classified based on their polarity as like in this figure. So amino acid divided into two two parts: hydrophobic amino acid and hydrophilic amino acid. So hydrophobic amino acid uh, in the it's under on non-polar. It's There are two parts, polar and non-polar. So polar is hydrophilic amino in the hydrophilic amino acids, and non-polar is hydrophobic amino acids. So non-polar is divided into two parts, are al- alkyl group and aromatic group. So in the part of alkyl group, glycerine and aniline, valine, neosine, isoleucine, methionine, toluene, and R मतलब aromatic group. So phylolenine, tetrapotopane, and hydro hydrophilic amino acid. They are polar, and polar are there are three types: basic, acidic, and neutral. So first of all, neutral in the tetrosine, serine, theronine, citrine, glutarine, asparagine, and acidic, glutamic, aspartic, and basic is lysine, itidine, arginine. So next slide is classification based of essentialities. So essential amino acids are amino acids which are need through your diet because your body can't make them. Whereas non-essential amino acids are the amino acid which are not an essential part of your diet because they can be synchronized by your body. So essential is histidine, isoleucine, leucine, methionine, phenylalanine, and etc. And non-essential is alanine, arginine, asparagine, aspartate, and etc. एक्सेट्रा बहुत सारे हैं. So peptide bonds, amino acids are in peptide bonds. Amino acids are linked together by amide group called peptide bond. During protein synthesis, the carboxylic group of amino acids at the end of growing polypeptide chain react with the amino group of an incoming amino acid, realize molecules of water. The resulting bond between the amino acid is a peptide bond. So you see that in this figure, um, it is a peptide bond, and it's incomplete of peptide bond. So next slide is the structure of protein. The sequence of a protein is determined by the DNA of the gen, the encoded of proteins, or that encodes a portion of the protein for multi sub subunit proteins, and change in a genes basically genetic DNA sequence may lead to the change in the amino acid sequence of proteins. Even changing just one amino acid in a protein sequence can affect the protein's overall structure and function. To understand how a protein gets its final shape or conformation, we need to understand the four level of proteins, like primary, secondary, tertiary, and quaternary. So primary structure, the simplest level of the protein structure. Primary structure is the simplest. The sequence of protein amino acid is a polypeptide chain. The hormone insulin has a two peptide polypeptide chain A and B. The sequence of A chain and the sequence of B chain can be considered as a example of primary structure, like in this example. So there is A chain and B chain. So you see that there is a S to S connect with another A to B chain. So we have to explain later in depth. So secondary structure refers as a local folded structure that form within a polypeptide due to interaction between atoms. The most common type of secondary structure are the alpha helix and beta replicate sheet. Both structures are held in shape by hydrogen bonds, which form between the car- car- carbonyl of amino acid and the amino H of another. In this structure, you can see that this figure, secondary structure. So a helix structure, example of amino acid subunits and B plated sheet. So there is a purple block in this figure. So this is a beta plated sheet. Another is a Ternary structure. The overall three-dimensional structure of the polypeptide is called Ternary structure. The Ternary structure is the primarily due to interaction between the R group of the amino acids that make up the protein. Important to Ternary structure are hydrophobic interactions in which amino acid and the non-polar hydrophobic R groups cluster together on the inside of the protein, leaving hydrophilic amino acids on the outside to interact with surrounding water molecules. So you can see the figure like this. There is a chain of polypeptide backbone. So it is attached by the hydrophobic interactions and hydrogen bonds like together an ionic bond also to connect the polypeptide backbone. And the next quaternary structure, when the multiple polypeptide chain subunits come together, then the protein attain its quaternary structure. There is the example of quaternary structure is the hemoglobin. The hemoglobin carries oxygen in the blood and it made up of four subunits, two each of the alpha and beta types. So there is an example of these figures. It's a quaternary protein structure. It's called क्या कहते हैं, hemoglobin. So I think that's it. And another point is demonstration of protein folding. Each protein has its own unique shape. It is a temperature of pH or a protein environment is changed, or if it is exposed by the chemicals, these interactions may be dispersed, causing the protein to lose its three-dimensional structure and turn back into an understructure string of amino acids. When a protein loses in a higher or order structure but not its primary sequence, it is said to be detonated. Protein are the usually non-functional, and these are the reference from we have to pick up the lines and uh, paste in the PPT. And another is image reference says, and you can also pick up. And the next, thank you, ma'am. That's it.